Kia ora, I'm Eleanor. Thanks for joining me for this presentation, Whakawhanaungatanga using DNA, MyHeritage. This talk is about how to find the matches at MyHeritage who are most likely to share recent common ancestors with you. It's a follow-up to a 3-part series I created for Roots Tech Connect, held online during February 2021. The series is still available online to view for free. If you haven't watched the Roots Tech series yet, I recommend that you pause this video and watch the Roots Tech series first, or at least watch part 1, which explains the basics of DNA inheritance, why Māori may face extra challenges when using DNA test results to Whakawhanaungatanga, and how we can start to overcome those challenges. There's a link to the series in the description below. If you follow that link, you'll also find a link to a glossary for the kupu Māori words that I'll be using in this talk. If you want to just keep watching this video for now, then that's all good too. I will be repeating some of the same info that is in the Roots Tech series. If you have questions at the end of this MyHeritage talk, you may find the answers in the Roots Tech series or later videos that will be appearing on this YouTube channel. Depending on your questions, you might also want to watch my talk at Legacy Family Tree webinars. This talk focuses on how Y-DNA test results can provide a very helpful complement to DNA test results from companies such as MyHeritage. This talk was free to view for the first week, but it now requires a subscription to view. Subscriptions are frequently on sale, so look out for a special if you don't yet have a subscription. Before we go further, I want to emphasize that this talk is not about me pushing DNA testing as a way to prove you're Māori. Although for those of you who aren't sure if you're Māori or not, because you don't know your whānau or whakapapa, perhaps because you or your parents were adopted, DNA testing may be able to help. This talk also isn't about privileging DNA testing over going back to your marae and learning about your whakapapa from kuia and kaumātua, if that is an option for you. Nor is this talk about convincing people to DNA test, and nor is it about supporting theories or laws based on blood quantum. These are all common and understandably important concerns when the topic of Māori DNA testing is raised, but I won't be discussing them further today. If you'd like to know more about these and other concerns that DNA testing may raise for Māori, you'll find a link to an article on that topic in both the Roots Tech series handout and the Legacy Family Tree webinars handout. So what are we going to be looking at in this talk? Well, we're going to be looking at using DNA testing as a tool for Whakawhanaungatanga. DNA test results can help us to make connections with the whanaunga and learn more about ourselves, our whānau, and our whakapapa. And of course, that includes learning about our non-Māori relatives and any non-Māori ancestors we may have. I'm going to assume you've watched at least part 1 of the Roots Tech series, and you know that because of the amazing history of Pacific migration, if you have one or more Māori great-great-grandparents, the matches who appear at the top of your MyHeritage DNA match list might not actually share recent common ancestors with you. You also know that instead of having a smallish number of close matches who share lots of DNA with you, you may have hundreds or even thousands of so-called close matches. Even though these matches do share lots of DNA with you, most of them won't actually share recent common ancestors with you. To find the ones who do, you'll need to sort through your match list. If you've watched the Roots Tech series, you'll know the matches to look out for. They'll be the matches who share many large segments of DNA with you. At MyHeritage, you want to look out for matches who share at least one segment of 40 centimorgans or more, and at least a few other 30 centimorgan or larger matches, larger segments as well. It's a bit of work finding these matches who are most likely to share RCA, recent common ancestors. But if you follow the process at the end, you'll have a customized list of priority matches. These matches would be at the top of your match list if the match lists were built for people from communities whose DNA test results are impacted by historical founder effect and endogamy. Of course, you're always free to make contact and learn about and work out your connections with any of your matches, but I recommend that you start with your priority matches in your custom-built match list if you're looking for unknown whānau and whakapapa. Let's look now at how MyHeritage DNA can help us to Whakawhanaungatanga. First off, many Māori from Aotearoa, New Zealand have tested at MyHeritage, so you've got a really good chance of finding matches in the database. You can sort your matches by largest segment and get access to the segment data, which is an important step in the research process. Many whānau Māori have family trees online at MyHeritage linked to their DNA test results. This information is going to make it easier for you to work out how you're related to your priority matches. Lastly, if you've tested at Ancestry, Family Tree DNA or 23andMe, you can get a preview of your matches at MyHeritage by transferring a copy of your raw data file to MyHeritage for free. If you find matches that you want to know more about, you can pay a one-time fee of New Zealand $45 per kit to access all the DNA tools and features. Or you can purchase a 6-month or annual subscription to unlock the DNA tools for all kits uploaded to the same account. Watch out for special deals. Sometimes there are free upload periods and the subscriptions are often on sale. If you are DNA testing to find whānau and whakapapa, I highly recommend transferring to MyHeritage because you never know where your best matches will have tested. Many of your whanaunga will have only tested at MyHeritage, but make sure you're comfortable with the terms of service before testing direct or uploading to any DNA database. A quick note, each company calls the largest segment something slightly different. This can be confusing. So just so you know, in this context, all of these terms mean pretty much the same thing. I'll do my best to stick to largest segment in this link because that's the term MyHeritage uses. Now let's take a look at an overview of the research process. First, we need to sort our matches by largest segment so we can quickly find the matches who share a largest segment of 40 centimorgans or more. Once we've found these matches, we need to check the segment data for each of them, so we can see the size of the other segments that are shared with the match. Next, we filter out all the small segments of DNA to get a new total that only includes the large segments of DNA. Then we can put that new total into the shared centimorgan tool at DNA Painter to generate more accurate relationship predictions. With these new relationship predictions, we are now in a better position to start looking at how we are related to our DNA matches. Lastly, we also have the option to work out the average segment. Working out the average segment can be a really quick way of gauging whether a match is likely to share a recent common ancestor with you or not. Let's now look at some examples from MyHeritage. Please keep in mind that the way that information is presented on DNA testing companies' websites is updated from time to time. So your results might look slightly different from the images you're going to see in this video. All matches' names are fictional to protect the identity of the tester and match. MyHeritage makes it super easy to reorder your match list. At the top of the match list, you just click on Sort By, and then in the drop-down menu, click on Largest Segment. Once the matches with the largest segments are at the top, you can start looking at each match one at a time. This is now the match at the top of the list. Let's take a closer look. On the left, we see the match is an estimated half-sister, first cousin, aunt, or niece. We can click on the little question mark to see some of these relationships on a family tree. Remember, these estimated relationships are based on the shared DNA. Other relationships are possible. For example, grandparent and half relationships, such as half sibling. Half relationships are not shown on this chart. Of course, most people never use the different terms shown here when talking about or to their whanaunga, but it is helpful to know which relationships these terms refer to, because DNA testing companies and online family tree programs do use these specific terms. Let's look back at our match again. We can see that the tester shares 17.8% of their DNA with this match, or 1,264.2 cm. cm stands for centimorgan, and it's a measurement of DNA. Now this total is really big, and we can also see that the DNA is spread across 55 different segments. The largest segment of DNA shared with the match is 171.6 centimorgans, and that's a very large segment. But how about the rest of the segments? How large are they? How many large segments are there? And is this match really a half-sister, first cousin, aunt, or niece? If we download the DNA info, we can see how much of the DNA that's shared with a match is in large segments. To get that info, we need to click on Review DNA Match. This will bring up a lot of interesting info, including shared matches. If you have a subscription or have paid the upload unlock fee to view, or have uploaded during a free unlock special time, you will see this kind of information. For today, we're going to scroll straight down to the chromosome browser so we can get to the segment data, and that's what we're looking at. Well, we're looking at the chromosome browser now. Now, the segment data is only available for your matches who have opted into sharing. Just like at Family Tree DNA, the chromosome browser at MyHeritage is not to scale. You can check the length of the shared segments by hovering over them. But what we want to do is download all the segment data. You'll find the place to download the segment data just above the chromosome browser. It's called Shared DNA Info at MyHeritage. For Māori and Polynesian matches, I recommend that you always check the Shared DNA Info to see how many large segments you share with a match. When you download the Shared DNA Info, you'll receive a spreadsheet file. You can open this in a program such as Excel or Google Sheets. You need to add up just the large segments to get an adjusted total. Let's take a look at an example. I've downloaded the DNA info for our match Kui, and these are all the segments that are 30 centimorgans or larger. Now, if we add up the total of just these segments, we get 773.8 centimorgans. Another way of adjusting the total is to copy and paste the segment data into the individual match filter at DNA Painter. I gave the full steps of how to use this tool in part 2 of the Roots Tech series, the Family Tree DNA talk. I'm just going to skim through the process now. So we just click on the box to go to the tool and follow the steps to get your adjusted total. And here we're at the end with the result. And this is good news. It's pretty much the same total as what we got when I did the maths myself. From here, we put this adjusted total into the Shared Centimorgan tool at DNA Painter to generate adjusted relationship predictions. Just a reminder, this tool is not designed to be used with matches that you know share multiple common ancestors and therefore multiple relationships with you, like your Māori and other Polynesian matches will. The estimated relationships could still be really far out when we use this tool, but we're using an adjusted total based on only the segments 30 centimorgans and up. So the predicted relationships for Māori and Polynesian matches are more likely to show the most recent relationship as one of the options. Just click on the green box to go to the tool. And then enter the adjusted total that you share with the match. Here I have entered the adjusted total of 774. Scroll down on the page when you're in the Shed Centimorgan tool and you'll see this. The predicted relationships when a match shares 774 centimorgans. There is a lot of info on this slide. What the info is telling us is that there is an 89% chance that the tester and match are related in one of the ways listed in this top box, and an 11% chance that the tester and match are related in one of the ways listed in the second box. Just a word of caution, some of the relationships have a dagger symbol beside them. This is because the info in this tool comes from two different sources. It gets a little bit technical as to what it all means, but for now, you can just focus on the relationship options that don't have a cross beside them. If you scroll a bit further down the page, you can see the possible relationships for you and the match in this coloured chart view. Now, if we go back and compare the estimated relationships that MyHeritage gave us with those from the Shed Centimorgan tool, we can see there is a difference in the possible relationships. The only relationships that appears on both sides is first cousin. The closer relationships that MyHeritage estimates on the left, half-sister, aunt or niece, are not highlighted as possible relationships in the Shed Centimorgan tool on the right. This is because the total we used with the Shed Centimorgan tool is 500 centimorgans lower, and that means some of the close relationships that MyHeritage suggested are just not possible. So just to restate, we have 1,264 centimorgans shared with the match at MyHeritage versus an adjusted total of 774 centimorgans at the Shed Centimorgan project. This match Kui is actually known to the tester, and the known relationship is first cousin once removed. Now that means the tester and Kui are closely related. They share a recent common ancestor. It's the tester's grandparents and Kui's great-grandparents who are the same. Several whānau members have tested, and all the relationships support that this is indeed the correct relationship for the tester and match. Removing the segments under 30 centimorgans from the MyHeritage total and then using the Shed Centimorgan tool to get estimated relationships has given us relationships that include the actual known relationship of first cousin once removed. 774 centimorgans is still considered to be quite a lot of DNA to share with a first cousin once removed. And that's why there is only an 11% chance, according to the tool, that the match actually is a first cousin once removed. But at least the closest known, and in this case the only known relationship, is now one of the predicted possibilities. And this is exactly what we're hoping for when we remove the segments shorter than 30 centimorgans from the total shared DNA that is given to us at MyHeritage. We want more accurate relationship predictions. The most important information to take away from this talk is that all the small segments under 30 centimorgans, which we removed, that's all of them here, they inflated the amount of shared DNA and meant that the predicted relationships were closer than what the actual relationship is between the tester and Kui. So just to be clear, this method of subtracting small segments and then putting the new total into the shared centimorgan tool won't always give you predicted relationships which include the actual most recent relationship. But it can help you get closer to what the actual relationship is. And I think it's worth trying. Let's take a look now at another example. This tester and match are estimated to be a great uncle, great nephew match, or perhaps a great grandson. If we click on the question mark, we can see these relationships in a family tree. Once again, MyHeritage is just showing us some of the possible relationships when you share this much DNA with a match. There are also other possibilities when you share 409 centimorgans of DNA with a match. For example, first cousin once removed, first cousin twice removed, second cousin, great-grand uncle or aunt, which is probably more commonly referred to in New Zealand English as great-great uncle or aunt. There are also half relationships which are possible, such as half first cousin. And once again, the half relationships don't show on this tree view. Now let's look back at our match. The largest segment doesn't quite meet our preferred minimum largest segment of 40 centimorgans. But let's look at the match anyway, because you might not have many matches who share a larger segment of 40 centimorgans. So if we download the segment data and remove the segments under 30 centimorgans, this leaves just 2 segments and a total of 71.6 centimorgans. The shared centimorgan tool gives us these relationships. There are a whole lot of options, and if you were wanting to work out how you were related to this match, you would need to actually check out all these options by doing thorough family tree research. Some options you might be able to rule out based on the age of the tester compared to the age of the match. The main takeaway from this is, once again, that the close relationships predicted by MyHeritage, based on the total of 410 centimorgans, I rounded it up from 409, are not options using the adjusted total of 72 centimorgans at the shared centimorgan tool. Now this match isn't known to the tester, but the tester and match do have pretty good knowledge of their whakapapa. They're from completely different iwi, although they are iwi that are sort of neighbouring iwi. Now both the tester and match's whānau have, like many members of both the tester and match's whānau have tested. So we can be quite sure that this match does not share a recent common ancestor. And it's possible that none of these suggested relationships by the shared centimorgan tool are accurate, and that the most recent common ancestors are further back in time. But either way, this match would not be a priority match if you didn't know your whānau and whakapapa, because it would be very difficult to identify, if not impossible to identify, who the most recent common ancestors are. If you don't want to have to deal with downloading segment data when you're wanting to learn more about your matches, you do have another option at MyHeritage. You can go to Tools, then the chromosome browser, click on Explore, and this will take you to the one-to-many chromosome browser. You need to know which match you want to compare yourself or your tester to before you use this tool. You can't sort the matches by largest segment on this page. That step needs to have been done before in your main DNA match list, and you need to have identified which matches share a largest segment of 40 centimorgans or more before you come to use this tool. When you come to the page, you put the name of the match that you're wanting to compare yourself to in the box here. I'm wanting to compare Haki to Iri. So I put Iri's name in this box and click on the little search symbol to search for her in the match list. I find her in the list, click on her name, and she'll appear alongside Haki. Then I need to click on Compare, and I'll get this page. I need to scroll down past the chromosome browser to here. Then I click on the drop-down arrow to the left to reveal the segment data, like this. I can now copy and paste this data into the individual match filter at DNA Painter and get the tool to recalculate the total. And then, like I've shown you, we take that total and put it into the adjusted, to the shed centimorgan tool at DNA Painter to get more accurate relationship predictions. Now, this workaround avoids using the Google or Excel spreadsheets, but you will still probably need to do all of this research on a computer. It isn't going to be easy or even possible to do it on most phones or tablets. Before I finish this talk, there's one more workaround that I want to take you through: how to calculate the average segment of DNA you and a match share. Average segment is another way of assessing how much a match is impacted by founder effect and endogamy. And if your match hasn't opted into sharing segment data with you, it will be the only workaround that you have. So let's go back to look at our match queue. The info we need to work out the average segment is here. We're going to take the shared DNA and subtract the largest segment, and then divide the remainder by the number of shared segments, -1, because we've subtracted the largest segment. So let's look at the maths for this. We have the shared DNA, 1,264 centimorgans. We're going to subtract the largest segment. That gives us 1,092 centimorgans. Next, we need to divide this number by the number of segments, (1). So we're going to divide 1,092.4 centimorgans by 54 segments. And this gives us an average segment of 20 centimorgans. Now this is a really good large average segment. The shared DNA is also high, and so we can say with confidence that the match definitely shares recent common ancestors with the tester. So this tester is one of our priority matches. Let's take a look at our other example. Let's do the maths for this match. We have the shared DNA, 410 centimorgans, and we're going to subtract the largest segment. That gives us 373 centimorgans. Next, we need to divide this number by the number of segments, -1. So we're going to divide 373 by 38. And this gives us an average segment of 10 centimorgans. Now, 10 centimorgans is quite a short average segment. Even though the total shared DNA is high, this total is made up of mostly small segments. So as we saw when we looked at the same match and removed all the segments under 30 and there were only 2 left and the total was only 71 something centimorgans. This match, going off the average segment, is unlikely to share a recent common ancestor. It's possible, but it's not that likely. And again, if you don't know your whakapapa, these kinds of matches who share short average segments will be difficult to work out the connection to. Now, most of your Māori and Polynesian matches will share short average segments with you because out of these thousands of matches that you'll have in your DNA match list, not many of them will actually share recent common ancestors with you, such as a great-grandparent or closer. If you've watched the third part of the Roots Text series, you would know that at Ancestry, I recommend looking for matches who share a longer segment of 30 centimorgans or more. Now, I learnt this 30 centimorgan minimum longest segment from the research of Kalani Mondoi, admin of the Polynesian project at Family Tree DNA and admin of the Polynesian DNA Facebook group. I add in an additional recommendation to look for matches who also have an average segment of 11 centimorgans. The research on average segments was first lectured about by Leila Pearl-Larkin in 2016. Leila is also known as the DNA Geek. I read about it in a blog post by Mercedes Bronze that was published in 2018. And a few of us in the Polynesian DNA project worked together to come up with a minimum segment, a minimum average segment, above which all our matches seemed to reliably share recent common ancestors. We've sort of changed things around a little bit since Ancestry started showing the longest segment. When we were first looking at this together, they didn't show longest segment. But now that they do, my recommendation is to do the maths like I've showed you for these MyHeritage examples and look for matches who share a average segment of 11 centimorgans or more. Now at MyHeritage, we're going to use the same average segment workaround. Well, I recommend that it's a good thing to use, but I recommend raising the minimum largest segment to 40 centimorgans and the minimum average segment to 15. These higher thresholds are to try and account for imputation, which has this tendency to fill in small gaps between multiple small segments, thus creating large segments that we don't see at other DNA testing companies. A future talk is going to explain more in detail on imputation. But for now, I recommend using these thresholds, particularly if you're looking for unknown whānau and whakapapa. Of course, you also want to share a good sized total as well, and 100 centimorgans is a good minimum. We're almost at the end of the talk and we've covered quite a lot, so let's just recap the process at MyHeritage. Currently, if you have one or more Māori or Polynesian great-great-grandparents, you will probably need to reorder your DNA match list by largest segment in order to identify the matches who share recent common ancestors with you. For Māori or Polynesian matches, remove segments under 30 centimorgans. Then recalculate the total and use the shared centimorgan tool to generate more accurate relationship predictions. As an additional or alternative workaround, you can calculate the average segment. At MyHeritage, you can add notes to your match list as you go, so you're only going through the maths once for each of your matches. If you can, compare any matches of interest at another database, such as Family Tree DNA. If you or your match tested at MyHeritage before March 2019, you won't be able to upload a copy of your raw data file to Family Tree DNA. You and your match might need to upload to GEDmatch instead or Tested Ancestry. And as always, before DNA testing or transferring raw data files, check that you're comfortable with the terms of service. As you work through your match list at MyHeritage, prioritise your research on matches who share a larger segment of 40 centimorgans or more and an average segment of 15 centimorgans or more. These are the matches who are most likely to share RCA, recent common ancestors, with you. Wherever you DNA test, remember that segment length matters. I can't 100% guarantee that you'll share a recent common ancestor with all your priority matches, but they are the matches who will most likely share recent common ancestors with you. I'm also not saying to completely ignore the matches who share small segments or smaller totals of DNA with you. But for the Māori and Polynesian matches who do share only small segments or a small average segment, it is going to be much harder for you to work out the common ancestors that you share with these matches, unless you and your match have a really good knowledge of your whakapapa. And even then, it can be a challenge. Of course, finding the priority matches is only one step in the research process. There are others. Step 1 involves carefully looking at your ethnicity estimate, because the higher your percentage of Polynesian DNA, the more your results are likely to be impacted by this history of founder effect and endogamy. And so the more closely you'll need to follow, or the more closely I suggest that you follow this alternative guide. Step 3 is important because, as we saw in part 1 of the Roots Tech talk and also in the Big Y talk at Legacy Family Tree webinars, the standard tools for grouping or clustering matches in order to identify common ancestors typically won't help you to separate your Māori or Polynesian matches into family groups. Step 4 is important because DNA test results don't stand alone. We still need to do whakapapa or other traditional genealogy research. Step 5 will help you with your research, but it's really important to not pressure anyone to DNA test at all. And anyone who you're asking to DNA test needs to be fully informed of the process and the risks that can come from DNA testing. I touched on step 6 in part 2 of the Roots Tech series, and I talk about Y-DNA in depth in the Big Y talk at Legacy Family Tree webinars. I've put a link to that talk in the description below. Lastly, we can all learn so much more from our DNA test results when we collaborate. Once you've done the research that you can by yourself, I encourage you to reach out to your DNA matches. If your whānau has surprise matches, I hope they'll be welcomed and supported to connect with their whānau and whakapapa. If you have already tested your DNA and you want to learn more about the other steps, please feel free to join us at Te Ira Tangata Māori DNA on Facebook and or at Te Ira Tangata Māori DNA Family Tree DNA Project. You might also want to join some of the other specialist DNA help groups on Facebook. Some of these Facebook groups, such as Polynesian DNA, are open to people who haven't DNA tested yet, but who are interested in it. And there are also other family tree DNA projects that you can join if you have DNA tested. There are links to these in the Roots Tech and Legacy Family Tree webinars handouts, and I've also put a few links in the description below. If you're interested in other resources for learning more about the impact of historical founder effect and endogamy on DNA test results in general, and what other strategies you could try to get the most out of your DNA test results, you'll find links to other resources in those handouts. Thanks for joining me for this talk. I hope you found it useful. Best wishes Best wishes with your research. Thanks also to everyone who has helped me with this presentation. Ngā mihi nui kia koutou katoa.